How does the human brain work? How do we make sense of the sheer complexity of billions of neurons firing at different rates all the time and in every behavioral experience? The brain is a computer, but nothing like the ones that it designed and built. The brain is a computer that evolved over billions of years of animal evolution, and we're still trying to get our heads around it. Our next speaker is Professor Danielle Bassett from the University of Pennsylvania, and she is ideally placed to deal with the brain from a whole functional organ perspective. Like Schrödinger, Bassett is a physicist who has turned her attention to sorting out some of the problems of biology. But unlike most of her predecessors who have attempted to do this, she's actually getting it done. She originally did her undergraduate degree at Penn State University before moving to the University of Cambridge for her Ph.D., where she was a notable rower. After that, she was a Sage Fellow at the University of California in Santa Barbara, which is a difficult place to leave, and then moved to the University of Pennsylvania to set up her own lab as an assistant professor. She is now an associate professor in the Department of Bioengineering there. She has received numerous prizes and accolades for her achievements thus far, including the MacArthur Genius Award. Her work on the nature of the human brain draws on mathematical models in order to explain and to identify networks within the brain that show different kinds of topologies in different types of activities, such as memory, perception, language, and how these networks are maladjusted in conditions such as schizophrenia. Her work can be described on this subject as comprehensive, prolific, clear, and I think the most coherent analysis of whole human brain function to date. Please join me in welcoming Professor Danielle Bassett.
Thank you very much, Tomas, for that kind introduction. I'm, like Professor Tai, I'm very excited to be with you all today to share a little bit of the work that we've been doing over the last couple years, but also to hopefully show you a broader picture of where I think this part of the field is going over the next couple years. And in order to set the stage for that and to give an idea of perhaps how complex systems research might help us to understand the brain, I wanted to start with a quote from Victor Hugo in one of his most famous novels, published in 1862, where he says, "How do we know that the creations of worlds are not determined by falling grains of sand?" And I think this quote sort of motivates the broader question of how many large things can actually come from very, very small things. And to understand this quote and put it in context a little bit more, I wanted to think about a sand pile. So in a sand pile, a single grain of sand, whether added or moved, can cause massive macroscopic changes in the shape of the sand pile by initiating an avalanche. But this type of large macroscopic change due to a tiny change in a small part of the system is not something that just affects mechanical systems. It also affects large-scale technological systems. So consider the power grid, for example. On an afternoon in September of 2011, a technician reconfiguring circuits at an electrical switchyard in Arizona prematurely cranked open a hand-operated switch, shorting out the southwest power link and sparking one of the biggest blackouts ever to strike North America, affecting 7 million individuals. These two examples motivate the larger question: how can such small events drive such large events? And the answer to that question is, in fact, pervasive connectivity. So it's the pervasive connectivity among little grains of sand that can take the small mechanical perturbation of the movement of one grain to cause a massive avalanche in an entire sand pile. And similarly, in the power grid, it's the connectivity between that single switch that individual opened to the rest of the system that allows for the propagation of surges and alterations in the technological demands upon the system that create the blackout. So it's this pervasive connectivity that allows for these really perplexing properties. So the study of systems that are like this, where a very small perturbation can cause a large-scale change, is the study of what are called complex systems. And the Oxford English Dictionary has a very terse definition of a complex system, which is that it is a system that is consisting of many different parts, but in which those parts are connected to one another. Now, complex systems can be found in many areas of science and technology, but I think it's my personal opinion that the most quintessentially difficult and I think pervasively interesting complex system is that of human thought. And when considering human thought, we naturally begin with its primary organ, which is the brain. And so you must all be a little bit confused here because, clearly, the brain is a wet, gooey organ that sits inside of your skull. It's a tissue, it's a volume, it has nothing in common with either a sand pile or a power grid, and certainly does not appear to be simply separated into individual units or connections between them. So why is it that the brain is a quintessential example of a complex system? I think that is a question that has been wrestled with for a while. If we actually look at the brain, probably the most common representation is not as a complex system, but is as a map, similar to a map of the United States or a map of the counties of Ireland or a map of the European Union. And the notion that the brain can be mapped out into individual regions that may code for a specific function actually has a long history in the pseudoscience of phrenology, where the architecture of the skull and the tissue beneath it is thought to allow us to identify regions that code for very abstract functions, including ideality, benevolence, firmness, agreeableness, hope, and even human nature itself. I think this is rather beautiful, but it is, after all, a pseudoscience, and what has been done over the course of the last couple of years is to turn that pseudoscience into an empirical assessment of brain mapping. So using both invasive studies in animals, as we've already heard from the previous two speakers, and non-invasive studies in humans made possible by functional magnetic resonance imaging and other techniques, we now can identify regions of the brain that support movement, that support vision, that support language, audition, emotion, or decision-making. And in fact, even more recent empirical measurements have also allowed us to separate the brain not just into these large-scale systems, but also into individual units that are involved in very, very, very specific cognitive functions. So yes, certainly the human brain at this very large scale is composed of units, but if I am going to claim that this is a complex system, then I need to be able to show you where the connections lie. So where are the connections in this system? And indeed, I think that is one of the fundamental challenges. Identifying these connections is one of the fundamental challenges of understanding brain function. And the reason is that in general, when we think about any cognitive function that you perform throughout your daily life, it is not necessarily associated with the function of a single brain region. Instead, there's a whole constellation of brain regions and their interconnections with one another, either structurally or functionally, that allow us to take information, to process it, to transmit it between the regions that are necessary to perform certain computations, and then drive our human behaviors. This complexity produces the rich cognition and behavior that's characteristic of us as humans. It also complicates the development of effective interventions when those cognitive functions go awry, and we'll talk about that a little bit later. But to put that same sort of notion in words from someone much more eloquent than myself, I'd like to quote Henry Poincaré in his Science and Hypothesis 1905, where he says, "The aim of science is not things themselves, as the dogmatists in their simplicity imagine, but the relations among things. Outside of these relations, there's no reality knowable." That notion that in order to understand almost anything, in order to build knowledge, in order to press the future of science, I think what we have to understand are the connections between the units that we know and love so well. So where are the connections in the human brain? The answer to that question has been really pushed forward at a rapid pace over the last decade, where we've been able to use a tool, a sequence of measurements from a magnetic resonance imaging machine. This particular sequence is called diffusion imaging. And diffusion imaging is a non-invasive technique that we can perform in humans. where it maps out the diffusion of water molecules inside of the brain. Now, water molecules are, in fact, constantly bouncing around in your brain, moving by Brownian motion. We can map out exactly where those bounces happen and what overall constraints exist on the pattern of diffusion. And by looking at those constraints, we can reconstruct the pattern of what are called white matter tracks, or nerve fibers, or bundles of neuronal axons that connect up one region of the brain to another region of the brain. And what's exciting about this is that we can create this map, or measure this map in individual people non-invasively, and the architecture of that map is starting to be provide information on what makes each of us the way that we are. Now, the critical challenge, once we have constructed this connectivity map, is to understand the messy web that it constitutes, and to parse how that impacts who we are. How do we actually study these wires and their architecture? How do we characterize their spatial patterns? How do we distinguish different parts of the picture that differ between each of us? There's really important, I think, groundbreaking work that has occurred from many labs over the last couple, probably the last decade, maybe 12 years, that has offered a powerful new framework for understanding that pattern of connectivity. And that draws on tools from what's called network science. So network science is a very interdisciplinary emerging field in science that allows us to study systems that are composed of many interacting parts and where those interactions are highly heterogeneous. So not completely random, not completely ordered, but somewhere in between, and where that heterogeneity in the architecture of the connectivity tells us something very important about how the system actually functions. So we can take the data on the left-hand side and map it into a network model on the right-hand side, and then we have a tool that we have an encoding of the data that we can interrogate mathematically. We can model, we can predict, and we can quantitatively characterize. So intuitively, the nature of, why would we want to do this, right? Intuitively, the nature of that connectivity tells us about where information can actually be propagated. And we can see that on the left-hand side just by visualizing these structural highways of the brain. But we can also see it on the right-hand side where we've modeled the flow of information or traffic over this large-scale structural pattern. So what's important about this is that it provides us a way of thinking about how and where information can be transmitted that facilitates our cognitive functions as humans. So studies of this type have started to identify the fact that small world architecture in these networks is extremely important, consistently identified in brain connectivity in humans, but also extremely important for understanding how information actually flows in this system. So in order to unpack this statement, I wanted you to focus on two different architectural continua. This top one here is where we go from very little clustering, or a random network, to very high clustering on the left-hand side. And what we can see is that as you move from right to left, this clustering in the networks supports information segregation and local processing. On the opposite side, we can think about shortcuts in the network. There are very few shortcuts here, very many shortcuts here, and that actually supports information integration. You can see by the majority of connections here are very long range, and therefore can pass information from one part of the network to another part of the network very quickly, allowing for information integration. What's really interesting is that the brain sits in this intermediate regime in these network models, where it has a significant amount of local clustering, allowing this segregation of information, and then it also has several shortcuts within the network that allow for information integration across the system. And that helps us to understand that the way that the large-scale architecture of the brain is organized facilitates a balance between information integration and information segregation to support our cognitive functions. But as we're thinking about that, I think it's also important to realize that the structural features I've just been talking about for information processing are not the only type of connectivity that's important in a human brain. The pattern of shared information in the brain is also something that reflects important architectural constraints and facilitations for cognitive function. So while structural connectivity remains relatively constant at this very large scale, the patterns of shared information, which I'm illustrating here, can change as a function of time. And understanding that dynamic connectivity can change and help us to understand how our thoughts change or meander over time, or help us to perform specific tasks that are in front of us. So when we consider these notions of dynamic functional connectivity, we can ask ourselves, what are some fundamental network substrates that can support the dynamics of adaptation and learning that are characteristic of us as humans? And I think that question, a sort of resounding refrain that has come from asking that question, is the notion of modularity, where if you have a system that can be decomposed into subunits, each of those subunits can be adapted or changed or moved or altered slightly in order to allow the system to change its behavior on a slow timescale, and eventually over very long timescales allows for something like evolution, for example. Well, on the top right, I'm just showing you the notion of modularity in structure and morphology in the beaks of Darwin finches allows for the changes over evolutionary timescales. Modularity of mind is also a notion that's been proposed by many individuals, the idea that there are cognitive processes that can be strongly separated from one another. And then there's also some emerging work in the field of network science that allows us to quantify and identify where modules might be inside of a network. And given the fact that this substrate could be particularly important for adaptation of a system, we ask the question, is the brain actually modular in its network architecture? And the answer is certainly a resounding yes. Consistently across multiple labs and multiple data sets, it's clear that the architecture of functional connections in the brain, both at rest and while individuals are performing cognitively demanding tasks, is absolutely highly modular. So on the left-hand side, what you see are nodes, or these spheres, that reflect single brain regions, and then the connections between them provide information about shared information between those two regions. And what you see is this strong clustering of connectivity within a module, and then not very strong connectivity between modules. Importantly, the color code on the left-hand side here is the same as the color code on the right. So what you can see is this set of blue dots here corresponds to these regions in the visual cortex over on the right-hand side. What you can see is that some modules are very spatially localized, like this one in visual cortex, while others are spatially distributed, like this red one here, which is distributed across very disparate sectors of the brain. Importantly, several studies have now confirmed that each of these modules seems to be composed of regions that code for similar functions, and that suggests that the architecture that we are seeing reflected in the network organization of this system tells us something about what different pieces of the brain are actually doing. Now, to motivate the next few scientific questions, I wanted to offer a short interlude from Alexander Pope, where he says, "There is some peculiar in each leaf and grain, some unmarked fiber, and some varying vein. Shall only man be taken in the gross? Grant but as many sorts of mind as moss." You might like to know that there are, in fact, 12,000 species of moss. Obviously, there are many more examples of brains, both male and female. And that motivates a lot of the questions that we've been asking over the last couple of years of understanding individual differences. So what makes one person particularly good at X and another person particularly good at Y? We've addressed that question by looking at this inherent modular structure and asking how it changes or adapts to different situations and different tasks. And what we found is that while modular architecture is consistently identified across a range of different timescales, there can be small reconfigurations. So notice here that across the range of time, this is just a schematic, there are still 3 modules, peach, yellow, and blue, but the actual architecture changes, and in fact, even some regions, like this one at the top here, can change their module affiliation, initially being involved with the peach module, and eventually being involved with the blue module, or the yellow module. We can quantify that by showing that the flexibility in this assignment of regions to modules changes as a function of time, and it changes differently in each individual. Some individuals have very strong flexibility of their functional modules, and others have less flexibility. We've shown, and others have shown over the last couple of years, that flexibility in these network modules predicts individual differences in visual motor learning, in cognitive flexibility, in working memory, in reinforcement learning, and also in positive mood. Very interestingly, more positive mood is related to higher flexibility. Now, That work has actually motivated much broader questions. So this paradigm of network architecture being a critical part of complex systems behavior has motivated questions in network computation. So when we think about network computation, how do we define causal drivers of computation in these networks with loops or small world architecture or modularity? How do we quantify changes in information that occur in this network and how that impacts other networks, and finally behavior? It's also motivated questions in network development. So when we view this system as a network, it changes how we think about how that network architecture might change as a function of time as we move from childhood to adolescence to adulthood and then into later age, and how that change in network architecture facilitates the changes in cognitive capacity that are associated with these various periods in our life. It's also really fundamentally changed how we think about pathology, both in neurological disease and in psychiatric disorders. So there's emerging work in computational psychiatry and neurology demonstrating that these disorders, from epilepsy and Alzheimer's disease to schizophrenia and autism, are associated not just with pathology in local areas, but in fact with pathology in a distributed set of areas that map onto the complexity of the symptoms that individuals display as they come into the clinic. And so there's very important work that's being currently undertaken to map out these network pathologies in each of these diseases, their development, maintenance, and change to better understand the complexity of symptoms that are actually produced. That leads me to the final area that I'll mention today, which is that in viewing this brain as a network, it actually changes how we think about interventions. So now, instead of thinking about intervening in a single brain region, so resecting an area of the cortex that may be involved in an epileptic seizure, or trying to titrate stimulation to a single brain region in order to mitigate the effects of major depression, for example, what we are now thinking about is that, in fact, what we'd like to have is a way of intervening on the network, knowing that single interventions will affect a broader circuitry. So there's important, and I think really cutting-edge work that blends tools and theories from engineering with empirical approaches from neuroscience to help us to understand how to intervene knowledgeably in these network architectures to reliably predict how to guide the system, both in clinical contexts, but also in terms of cognitive enhancement. So that brings me to the question of what does the future hold? I think in the context of complex systems, the really cutting edge work that's being done right now is to extend our tools from a single region or a single network scale to multi-scale, multi-layer, and multiplex networks. And I think the most obvious place to start doing that is by taking what we've been doing at the regional scale and moving it down into the neuronal scale. So it's clear that large-scale brain activity provides just a coarse-grained encoding of the neural processes, and the map from these cellular dynamics to the regional dynamics reflect important constraints on system function. So by combining these two layers together, we can ask, how do cellular processes actually shape circuit behavior? But I don't think that that's where we should stop. I think we then need to go even farther back to molecular networks. And by understanding those molecular mechanisms, we can understand how they may impact large-scale brain network function. And that's going to be very important in developing effective pharmacological interventions. By extending this network modeling approach down through multiple layers like this, we can ask the question of how do genetic material and epigenetic drivers shape circuit behavior across spatial scales. But I also think that in a final extension, it's time to move up, to combine information from connectivity within a single individual, as I've shown here, to the social network. So while brain activity and structure offer important biological mechanisms of human behaviors, social networks offer important external inducers and modulators of those behaviors. So by extending the network model across these scales, we can start to ask the questions, how do brains actually shape the social networks that they are a part of? How does your brain change the architecture of your social network? And how does your social network impinge on the architecture and adaptation of your specific brain? This extension will be important in understanding human behavior within the context of broader culture and society. And now I'd like to leave you with One final thought. I mentioned a few slides ago that I think that the most quintessentially difficult and pervasively interesting complex system is human thought, but in fact, most of what I have talked about is the organ of human thought, or the brain. And now I think that it's important that the focus of our work is not just at the level of the brain, but extends into thinking about thought itself. And to do that, I think we have to face the fact that by studying the brains, societies, and cultures of today, we can fall victim to the ineffectuality of temporal primacy. So here is the human brain on the right-hand side, which exists today in all of its multicolored glory across each of us individuals. However, by studying just that one part of space, I think we confine ourselves to a very narrow window into the workings of the human mind. If instead we go outside of today, we are offered the rich window into the human mind that's located in literary work and in philosophical expositions, which can now be quantified with emerging tools from natural language processing, from network science, and from applied mathematics. And this added dimensionality of information can allow us to supersede the constraints of temporal primacy and provide a richer description of human thought. And I think that that will be important. I think it calls for a concerted effort to break down the current barriers between the sciences and the humanities. I think it also offers a call for a commitment to enhance precision in our descriptions of human thought itself, a call to enhance commitment to precision in our tasks constructed to probe human thought. And I believe that we can meet this call by informing our study of the brain with quantitative study of historic uses of natural language in literary work and in philosophical discourse. And in the words of a well-known Irishman, I have the words already. What I am seeking is the perfect order of words in a sentence. You can see for yourself how many different ways they might be arranged. And I think that by understanding how words are arranged and the meanings they reflect in historical literary work and in philosophical exposition, we can fundamentally reshape the questions that we ask about neural circuits in a profoundly new way, expanding our understanding of this quintessential complex system that is human thought. With that, thank you very much for your attention.